All right, so I don't think there's too much housekeeping today. We do have a special presenter later in the day, and I believe there'll be a book signing session later on if anyone's interested in that out in the foyer. But with that, we'll launch into the session for this morning. So I'm pleased to be chairing this session, which looks at the conservation of threatened species and threatened communities. And we've got a really nice panel here to show off the capabilities of botanic gardens in this space and the really great work that's happening in botanic gardens. So our first speaker for today, our keynote speaker, is Dr. Nathan Emery. Nathan is a Restoration Biology Officer at the Royal Botanic Gardens, Sydney, based in the Australian Plant Bank, which is part of the Australian Institute of Botanical Science. Nathan's research is a combination of plant ecology, seed biology, and restoration. He has worked with difficult to propagate species such as Actinotus and threatened Persunia, and has led several threatened plant translocation programs. He also works on the seed biology and conservation of threatened ecological communities in northwest New South Wales, with a focus on understanding the tolerance of seeds to temperature and moisture stress. And a big thank you to the Australian Institute of Botanical Science for supporting Nathan's keynote today. So thank you, Nathan, and welcome you to the stage.
Thanks, Lydia. Just put a timer on for myself too. Well, good morning, everyone. I'm really excited to be presenting you with kind of a little bit of an overview of the research that I've been involved in at the Australian Botanic Garden for quite a number of years now on threatened species, and more recently I've worked on threatened ecological communities in New South Wales. So at the Australian Plant Bank, we love Persunias. We've been working on them for many years, and we've come to the conclusion that they have really great persunialities, much like the scientists that study them. It never gets old, sorry. And for me, I've worked on a number of projects at the Botanic Gardens. The first project I was involved in was a recovery program for the critically endangered North Rothbury Persunia in 2013 to 2017. In this project, we were looking at ways we could translocate, or how to translocate this species in order to increase numbers of it in its very small distribution. And to get some better outcomes with this, we looked at its in situ and ex situ research actions, including seed biology, looked at developing propagation protocols, looked into the reproductive ecology of the species, and with our translocations, we were able to put almost 1,000 additional plants into the landscape, both within its known distribution and also outside of its distribution. More recently, our genetics team, Restore and Renew, Myrtle and others have looked at the population genetics of this species, which has been the motivation for a kind of a stage 2 of this project, where we're conducting a larger scale translocation of the North Rothbury Persunia, which is guided by those genetic results. The other project I've been involved in is an ACARP conservation project. So this is coal mining funded project. Initially it was looking at propagation protocols of a number of threatened Persunias in New South Wales, but has since been sort of narrowed down in its focus to Persunia hirsuta and Persunia hindii, two endangered species that occur on mining lease. Again, like the North Rothbury Persunia, we've looked at a number of different aspects, a number of similar aspects into the ecological requirements of these species, as well as a few other things like habitat surveying and modelling. We've also done some in-depth analysis of the soil environment, and more recently we've also started looking at population genetics, and as well as conducting experimental translocations. A younger project that I've been working in is the Relict Rainforest Woodland Conservation Project. So this is looking at 3 threatened communities in the Northwest Slopes in New South Wales. And in this project, we're combining in situ monitoring with seed biology research in order to understand the reproductive niche of the species within these communities. Okay, so let's talk Petunia because that has been my long-term passion. If you're not familiar with this group of plants, they're part of the Proteaceae family. They have small hermaphroditic flowers in contrast to some of the other more common Proteaceae that you may be familiar with, like Banksias, for example. Their dispersal unit is a fleshy drupe which contains a hard woody stone which protects 1 to 2 seeds within it. There are 99 species of Petunia endemic to Australia, all of them, and 9 of these species are threatened, listed as either vulnerable, endangered or critically endangered, 8 of which are in New South Wales. And the sad reality is all these threatened species continue to decline throughout their distribution. We've talked a lot about threats, key threatening processes in this conference, land clearing in particular, and that's no different for these Petunias. Their habitat's quite fragmented, populations are very isolated due to continual urbanisation in the Sydney Basin region as well as through mining. But there are also a number of other threats for these species, including grazing, slashing, predation, and also inappropriate fire regimes. And this has not been helped by the fact we have poor species ecological information of many of these threatened Petunia species. They've been hampered by very low recruitment success. Recruitment's rare in the wild. They have a very complex germination strategy and highly inconsistent germination results, which has hampered the production of plants for things like translocation. So these are what we term as exceptional species with complex requirements. So many of these species have a translocation action assigned to them, which necessitates that populations be supplemented or that new populations be established in order to build resilience for these species. And in the case for Petunia hirsuta and Petunia hindii, there's also a need for, in the mining industry, for these species to be incorporated into their rehabilitation programs. Historically, they've been unable to include them due to the issues surrounding understanding their ecological requirements, how to actually grow the species as well. But translocation, as we all know, putting plants into the ground is the easiest part of the whole process, but there's a very long a period of time before that, long lead up, that we need to better understand their ecological requirements. So some of the things that we've looked at for these species are trying to understand more about their habitat requirements, their reproductive ecology, things that might limit their seed production, seed biology, developing propagation protocols, and developing a translocation strategy for these plants. So we've conducted this conservation research in kind of a dual stream approach with in situ research actions and ex situ research too. The structure of these have kind of been a little bit fluid depending on the seasonality, when the seed is available for us to do seed biology research, for example. But there's been a multitude of research topics that we've covered, and I'm not going to have time to go into all of them today, so I've just cherry-picked a few that I feel are very important to highlight. But I also want to draw your attention to, in the middle, but because concurrent to this research, we've also run a series of iterative small-scale experimental translocations. And there are a couple of reasons for this. One, because we do want to understand how to best plant these species in the wild, but also to conduct them experimentally and to not wait until the end of our research before we just start looking at translocations, but to incorporate some of the research outcomes that we've attained along the way and implement those into our subsequent translocations and see what effect these actually have on the plants in the wild. So the first thing I'm going to talk about is habitat suitability. So we're going back to the basics and looking at the plants in the wild and trying to identify factors that might be associated with healthy plants, healthy populations. So we're basically saying if we've got plants that are doing well, they're healthy, they're reproducing, then this habitat may be highly suitable for these plants. So the things we survey for include vegetation type, associated species, as well as other co-occurring Petunia species, disturbances around the plants and in the local site, as well as the fire history of the site and understanding more about the properties of the soil. And so this was recently done by a PhD student, Sam Andres, who's recently submitted her thesis. She did this for Petunia hirsuta, went out across the distribution, surveying populations and their associated habitat. And there are a number of things that came up as being potentially important factors for the health of this species and the suitability of habitat. These included the abundance of Phytophthora-sensitive species in the area, so we don't actually know whether Petunia hirsuta is susceptible to Phytophthora. That work still needs to come. But plants, we found that plants were less healthy in areas where there were a high abundance of these Phytophthora-sensitive species in the habitat. Another factor that came up was the fire history of the site was an important consideration when looking at suitability of habitat. Sites which had no fire a couple of decades or more since the last fire, plants were not doing as well at these sites relative to others where there was a more recent fire. And there are a number of soil factors too that came up as being important: soil nitrogen, phosphorus and carbon, which were associated with population condition. So this is great. We can take this information and now extrapolate it and start looking at predictive modelling to look across it at the distribution and the landscape beyond. So we can do this via species distribution modelling, which was so really well sort of outlined and described at our workshop yesterday afternoon. So this is looking at habitat suitability of a species using traditionally using climate factors, but from our landscape work that and survey work that Sam did, we can include some of these key factors that we found to have potential importance with Petunia health. So these things, we can include things like the fire history of the site. We can also include species traits such as their range and also their dispersal mechanisms. They've got this fleshy droop, which 99% of the dispersal of those are through gravity. And in this study, we actually examined, or Sam actually examined, I should say, a number of threatened Petunia species as well as common species in the Sydney Basin region. And regardless of their listing status, we found that all 8 species that we investigated were predicted to lose considerable suitable habitat in the future. But this is also a useful way for us to start exploring the habitat requirements of these species. We can identify areas that may be climate refugia for these species moving forward when we're looking at areas for potentially translocating these species in the future. It also gives us areas to look and survey for potentially new populations because they've got high levels of predicted suitability of habitat. Okay, reproductive ecology. This is also really key for many Petunias. They've been found to be preferential outcrossers. So we wanted to study this in our threatened Petunia species that we were investigating to see whether this may be potentially a causal factor for recruitment failure or rare recruitment events. Also might be limiting seed production if these species are reliant on pollen vectors. So for this work we do pollinator surveys. We also do hand manipulation pollination experiments where we hand pollinate flowers using outcross pollen or using self-pollen, and then we bag those flowers to prevent pollinators from accessing them. And what we found with our species, like many other Petunias that have been researched, they're pollinated by bees. Maybe no surprise considering their small flowers. Mostly by native bees, particularly from the Leoproctus group, which is a small black bee which pollinates through buzz pollination. Importantly too, it's not a one-size-fits-all. We can't generalise with pollinator community. There is quite a bit of variation in the diversity and abundance of these potential pollinators across different sites. So it's really important to not treat each site as the same. And importantly, these species are preferential outcrosses and show some degree of self-incompatibility, which means that flowers that have self-pollen are less likely to produce viable fruit relative to those flowers that are pollinated using outcross pollen. So this moves on quite nicely to seed biology, which I've kind of mentioned early on has been a real limitation and a real sort of head scratcher for understanding the germination requirements for this group of plants, and in order to actually start establishing propagation protocols for growing plants from seed. So as I mentioned before, we've got a fleshy drupe, and within those, that fleshy exocarp and mesocarp layers, we have a hard woody stone, which is the endocarp, which protects the seed. And this endocarp needs to break down, it needs to be removed in order for the seed to have the potential to actually germinate. So when we're not in the lab, this endocarp breaks down over time in the soil, depending on the wet-dry cycles. This can be more rapid at times. But really what we've found is that it will break down slowly over time. So this is a graph of endocarp weakening during a soil burial trial for 2 years. Here the endocarp is weakened by more than 50% over the 2 years, but we're still not seeing in situ germination from these seeds, that despite the fact that the seeds are still viable and we're still able to germinate them in the lab when we remove the endocarp. And just to put this into perspective too, so on the y-axis we have penetrometer pressure, which is measured in newtons. So for fresh seed in February 2016, we have endocarp strength or pressure required of about 450 newtons. That's the equivalent of 40 to 45 kilos of pressure required before these endocarps will crack. And they're not actually that big, as you can see from the cross-section, but they are quite hard. Once the endocarp breaks down, the seed in many species has a physiological dormancy requirement that requires specific temperatures for germination to commence. So here on the panels, the graph panels on the left, we did a seasonal germination trial for Petunia hirsuta and found higher germination, or got higher germination when seeds were incubated under warmer temperatures in summer and also in spring and autumn. And we can take this a step further, kind of what I alluded to in my talk yesterday with a thermogradient plate. We can put these seeds on, look at their germination between these seasonal temperatures, and make predictions of when we might see the highest germination in the actual field site. And for here, for Petunia hirsuta population, we're getting a prediction of germination most likely occurring in the warmer summer temperatures. Now, because we wanted to conduct translocations very early on in the piece and not wait to the end, and the issues that were surrounding seed biology and understanding how to propagate from seed, we tried to fill in some of the numbers or get the numbers that we needed for these early experimental translocations, also using vegetative propagation. And this, like seed biology, has been quite a complex and difficult mechanism or process to understand how best to propagate these species from cuttings. So we got some cuttings and we ran a series of optimization treatments using various root hormone stimulants and looked at their impact on survival and strike rate of cuttings. And I guess what I really want to draw your attention to on this slide is the hazards ratio plot on the right-hand side, which is looking at the probability of survival of cuttings under different root hormone treatments relative to a water control. And down the bottom we have an Easy Root and Clonex purple treatment. Is the most potent chemical root hormone treatment that we treated the cuttings with, and we've got a significantly higher likelihood of cuttings dying before they actually strike roots in this process. So we're seeing a toxicity effect here of this root hormone chemical, which we want to stay away from, so that we give these cuttings enough time, which is sometimes up to 12 months for these cuttings to strike roots, so that we can maximize strike rate success. get there eventually. So let's talk about experimental translocations. As I've alluded to, they're concurrent to ongoing research, they're small scale, they're iterative, they're scientifically, experimentally designed planting events designed to try and develop optimal planting strategies for Petunia, as well as to include try and include some of our research outcomes as they come to hand wherever possible. So we're identifying best planting techniques for survival and growth, and as I talked about seed biology and propagation from seed and propagation from cuttings, there's a lot of information in the literature about which prop type leads to greater success in your translocation. So we had some plants propagated from cuttings. We also had some propagated from seeds. So we wanted to look at what prop type leads to the highest survival, at least in the short term. And for a very early Petunia hirsuta trial, we planted out 50-50 seedlings and cuttings grown plants. And we found that after 20 months of monitoring, 72% of survivors had a plant guard around them, so this helped to minimise the herbivory impact at the site, as well as provide some shade for these plants. This trial was commenced in 2019, which means that the plants were exposed to those extreme summer temperatures that coincided with the Black Summer bushfires. And that mulching was also beneficial for the survival of these plants, and we attributed this to a potential decrease in moisture loss from the soil directly around the plants. Now, when it came to looking at prop type, it was very, very interesting. All but one survivor were seed propagated plants. So we got some of the cuttings grown plants and some of the seedling grown plants, removed the soil from them, scanned their root system and ran some analysis to compare between the two prop types. And what we found was that the seedling roots showed a much, much greater root volume, area and diameter than what the cuttings propagated plants did. The seedlings had a lot more lateral roots. They were able to access a lot more moisture in the soil than the cuttings were, and that may have attributed to the result of seeing seedlings surviving far, far greater than cuttings grown plants. The cuttings themselves, at least at this stage early on in their life, they were putting more biomass into their above ground shoots. So we could say, you know, for Petunia, use seeds rather than cuttings because you're going to get a greater success rate in your translocations, but it's not always the story. And indeed, if you're growing your plants and choosing your plants based on certain genetics, certain genotypes, cuttings is obviously the way to go. So for Petunia, we're not discounting cuttings completely. We're just saying that from this result, we need to actually take into consideration that we need to hold these plants back longer in the nursery in order for them to start developing a larger root mass in order to increase their survival when we actually translocate them in the wild. And we saw this in a Petunia pauciflora trial where we planted out cutting grown plants that were 5 years old in the nursery, as well as seedlings which are only 1 year, and the cuttings far outperformed in terms of survival and growth than the seedlings. The seedlings did not do anywhere near as well. The other benefit of using cuttings, of course, is due to their nature. They will also flower and fruit a lot earlier than seedlings, which obviously need to go through their juvenile period, which for Petunia can be quite long. Okay, so I really want to just highlight a few really great outcomes that we've achieved through our conservation program for Petunia. One is the identification of new populations in the distribution. So being able to look at the suitability of habitat, go out and do some targeted surveys. We've been able to find new populations that are healthy, that are thriving. They're new sources of genetic diversity. They have seed that we can use and conduct in our experiments and grow plants on for translocation. So that's been a real success story out of this work. We've also been able to develop and standardise propagation protocols for Petunia for both cuttings and seeds. We're also working on tissue culture for this group of plants too, and we're making this information available to growers and people who want to be able to propagate this difficult group of plants. It's still a laborious process, but we're still getting consistent results from these protocols. Also highlighting the need to consider species interaction. So Petunia, you need to have the pollinator community present at your site, whether it's an extant population, a wild population, or a translocated population. It's no good having the plants there if you don't have the pollinators there who are going to promote outcrossing and promote those recruitment events, the build-up of viable seed in the seed bank. We've also been able to optimise planting to promote survival and success of our translocated populations of Petunia, so we have a number of recommendations for that. And importantly, with doing iterative small-scale translocations, not waiting to the end, we really wanted to make a lot of these translocations kind of a community engagement and interaction, an interactive event, get people involved in the planting. And so Petunia pauciflora in particular, the community really came together and were really keen to know more about this plant. It's called the North Rothbury Petunia. It's only found in their town, so they feel like they have some sort of ownership and want to know more about this plant. And involving them in the process creates this sense of stewardship for them over the plant and is really going to help maximise and ensure that this species persists into the future. And finally, we've had such a really good success from propagating these plants, in particular Petunia hirsuta, that we actually have surplus plants to what we initially required, what we set out to produce for our program. And this opens up opportunities for new collaborations, additional translocations, and this is what we're planning to do with this species. We're working now with Campbelltown Local Council and Kentland Bush Care Group, thank you, to conduct additional conservation translocations of this species in areas in the LGA where it has occurred historically. And we can do this with some confidence because we understand the habitat requirements. We know we need to survey the areas for pollinator presence. So this is a really great kind of add-on value to the work that we've been doing. Cool. So in the last 4 or 5 minutes, I'm going to quickly shoot through the younger project and take this up a notch and talk about conservation at the community level that we've started looking at in the Brigalow Belt South region in northwest New South Wales. So the 3 communities we've been looking at is Brigalow Woodland and 2 dry rainforest communities, semi-evergreen vine thicket and Cadelia pentastylis forest. Like threatened species, these communities also have a lot of knowledge gaps that are hampering effective conservation outcomes and ensuring that these persist into the future. For these communities in particular, the reproductive niche of many of the key species within these communities is unknown. And this is really important because climate change is a key threatening process for the region and for the communities. You can see the predictions there of a temperature increase of 1.5 degrees, lower winter and spring rainfall, and an overall reduction in soil moisture due to that reduction in rainfall. So for this work, one of the main streams is doing in situ monitoring at these communities. We have long-term monitoring plots set up where we're recording flowering and fruiting of plants within it. And we want to build up a data set that we can start to look at the reproductive status of these species and link it with temperature and rainfall. So we really want to answer the question, do some species within these communities only flower and fruit following specific climate events, or are there other species that are a bit more generalist? They tend to flower year in, year out, and they're not really like aligning with a certain, say, rainfall event. And it's been quite fortuitous to have this project set up in 2019 during, I could say fortuitous in hindsight now, because it was set up during at the peak of the drought. So we monitored these populations when they looked like they did on the left-hand images there. Fast forward to more recent with a couple of years of good rainfall, and we've seen that boom cycle return to these communities where abundance is and diversity is shot through the roof, and a lot of species are starting to reproduce. Alongside this monitoring work is seed biology research. So we're doing this work on key indicator species across the communities. The first aspect we're looking at is how they respond to temperature. So we're running these through the thermogradient workflow that I talked about yesterday, so I'm not going to go into detail about it. And this will start to build up a data set of species that may be that are predicted to have a potential germination seasonality shift in the future. The other side of the coin is understanding how these species respond to increasing water stress, so understanding their sensitivity. And we can replicate this quite consistently in the lab using a polymer called polyethylene glycol. It's a polymer that binds to water molecules and prevents them from the seed accessing them and imbibing them. So the more polyethylene glycol you have in your solution, the less water that's available to the plant. This is calculated as megapascals or MPa value, and we incubate the seeds at their optimum temperature that we've identified from the thermogradient plate and incubate them under a control, a low, medium and high water stress environment. And from this result, we then try and standardize this so we can make comparisons between species by calculating the MPa value where the final germination is 50% of the control treatment, and that's how we classify species as and their sensitivity to water. So we're slowly building up a data set of species that are common and key species in these communities, and we can provide this information in the first sense in a series of workshops that we've got planned for the region later on, provide this information to landholders, to nurseries, to relevant stakeholders, key parties, and also then provide feedback and create a discussion about what other things we can potentially look at that's important for these species. So I will just finish by saying obviously there are limitations to this approach. What happens in the lab isn't necessarily replicated out in the field. It's a lot more dynamic and unpredictable environment. So we really want to start ramping this up, look at more provenances of these species, multiple years, and also try and back this up too with some direct seeding field trials to start looking at those comparisons between the lab and the field. And I'd just like to thank everyone that's been involved in all this work that I'm representing here. It's certainly not an individual performance by me. It's been a massive team effort from the plant bank teams, the botanic garden teams, and as well as other industry and students too. So thank you.
Thanks, Nathan. Nice to see some personality. That's added a good taste to the morning. Our next speaker is an online presentation, Stephanie Chen. She's a PhD candidate at UNSW Sydney and Scientific Officer at the Australian Institute of Botanical Science, Royal Botanic Gardens and Domain Trust. She develops and optimises genomic resources for plant conservation. Thanks, Stephanie.
Hello, I'm Stephanie, a Scientific Officer at RESA at the Royal Botanic Garden Sydney and a PhD candidate at UNSW. Today I'm going to talk about opportunities in the genomic era for conserving myrtle rust impacted species. Genetic diversity plays an important role in the survival and adaptability of species and populations, and it's recognised as a key component of biodiversity. It enables a species to respond and adapt to a change or a challenge. And that's outlined in a chapter of the recently released germplasm guidelines. And there I also provide a case study on using that genomic information to conserve rust-impacted species. A key component of conservation and restoration genomics, and also understanding diversity, is sequencing. It's estimated 2 in 5 plant species are threatened with extinction, and the ongoing rate of plant extinctions is up to 500 times the pre-Anthropocene background rate. Over 90% of over 22,000 Australian native vascular plant species are endemic. and there are almost 1,400 plants listed under the Environment Protection and Biodiversity Conservation Act. So how can genomes help? Studying genomes can generate fundamental new knowledge of plant organisation, evolution and functions, which allows us to understand and conserve biodiversity. With rapidly decreasing sequencing costs and technological advances, more and more plants have been sequenced since Arabidopsis in 2000. However, sequencing efforts have concentrated on crops, their wild relatives, and model species. Therefore, the distribution of species with sequenced genomes across the plant tree of life is highly uneven. Myrtle rust is a plant disease caused by an invasive fungal pathogen first detected in Australia in 2010. It has a global host list of 480 species in Myrtaceae, and that's a key and dominant plant family in the Australian landscape. Myrtle rust cannot be eradicated, and detailed genetic information needed for an effective and coordinated response encompassing conservation management is lacking. So I've been involved in generating genomic resources for a number of these species, and those focused species have varying levels of resistance from highly susceptible to variable. So the general workflow we use is presented here. And kind of the idea is that we use this genomic information to optimize collections and conservation populations once we do those conservation actions to make those collections adaptable and adapted. So I'll be talking about work done as part of the Saving Our Species program, which is the New South Wales government's flagship threatened species conservation program. So I'll showcase the potential of genomic data in facilitating the management of at-risk species through optimizing ex situ collections. Today I'll focus on 2 rainforest species that are critically endangered due to myrtle rust, Rhodamnia rubescens on the left there, and Rhodomyrtus sidioides on the right. And both of these species are designated to the emergency priority in the Myrtle Rust Action Plan, and Rhodomyrtus is amongst the 100 priority species in the Threatened Species Strategy. So 281 ramets of Rhodamnia rubescens and 191 ramets of Rhodomyrtus sidioides were sampled across New South Wales by Craig Steen and RISA. This encompassed 43 sites for Rhodamnia and 34 sites for Rhodomyrtus. For wild collections, observations of rust infection and population information were recorded using the Myrtle Rust Proforma established as part of the New South Wales SOS Myrtle Rust Emergency Response Project. And so this is a photo I took in Wallingat National Park a few years ago, where I was collecting Rhodamnia, and you can see there's some plants you can see that have been infected in previous seasons. There's some suckers there that can be collected for cuttings, and also you see things like dead stands of dead trees. And so we filled out the Proforma, and that's just the kind of an example of what that might look like, information on rust, damage, as well as other traits of the tree. So the aims and objectives of this research were to describe genetic health, population structure, and genetic diversity across their known distribution in New South Wales, determine locality where it is unknown for plants in the ex situ collection, screen the ex situ collection at the Australian Botanic Garden Mount Annan and Booderee Botanic Gardens, which is in the Jervis Bay Territory, for clones to inform collection management, compare and determine diversity and kinship among in situ and ex situ plants to elucidate how representative the current ex situ collection is. And as an example of the possible applied interpretations of the newly available genetic knowledge, we determine optimal selection of individuals across several cryopreservation scenarios. So on to the results and recommendations. Genetic variability is present in Rhodamnia rubescens and is distributed along a latitudinal gradient, the same gradient seen in Rhodomyrtus. Maintenance of metadata and records underpins the usefulness of the collection, but genetic data was able to infer the geographic origin of plants where this information was unknown or lost, and that's represented by the grey dots in the PCA plots. So for example, there's a grey individual there of Rhodomyrtus, and that individual clusters most closely to sites which are in close vicinity, namely Hannam Vale, Crowdy Bay and Blackhead. So we can say that it's likely that the individual originated from that area. The genetic diversity of individuals with unknown locality is already represented by other plants with known and complete metadata, and therefore do not need to be prioritized. We calculated F statistics, so observed and expected heterozygosity as well as the inbreeding coefficient, and that indicates a preferentially outcross mating system for both species. Both species distribution extends north into Queensland, and from the observed patterns, it's expected that those Queensland populations harbour genetic variation that's not represented within the current ex situ collection. Therefore, sampling and analyses from Queensland are recommended to identify and capture increased genetic variability. A genet is a genetic individual, and a genet can be made up of multiple ramets or clones. For Rhodamnia, there are 40 unique genets among the 80 individuals from ex situ collections genotyped. These individuals included 24 from Bouderie, 39 from the Mount Annan ex situ collection, and 17 from tissue culture. Genets of multiple ramets were identified across each of these collections. There was minimal overlap between the three collections. Only one genet was shared. For Rhodomyrtus, of the 191 samples genotyped, 98 were from the Mount Annan ex situ collection. 60 unique genets were identified, and 18 of those genets are represented by multiple ramets. This information can be used to streamline ex situ collections, and redundant clones can be distributed to other botanic gardens or used for rust experiments to determine if certain genets are more resistant to rust. Field observations can be confounded by environmental factors, so don't give an accurate indication of rust susceptibility, and gathering that information from different data streams, genetic, field phenotype, and experimental phenotype, is valuable in building capacity for an effective conservation response. The next thing we looked at was, are the ex situ collections representative of wild diversity? The answer was yes for both Rhodamnia and Rhodomyrtus, which is good to see. However, the Rhodamnia, Mount Annan and Bouderie botanic gardens represent different parts of the species genetic diversity. So Bouderie is more representative of the southern populations. So strategic mixing of material from both collections is desirable for maximising genetic diversity in conservation actions. So now we have all this genetic information. How do we translate this into outcomes for conservation? Here we provide an example for optimising the selection of individuals for resource-intensive methods such as tissue culture or cryopreservation, and that's happening at Plant Bank. And it's particularly suitable for these species, as with many rainforest species with fleshy seeds, which cause problems for seed banking. So it's strongly discouraged to randomly select individuals for a conservation population, as this will diminish the long-term survival capacity of populations. We implemented an algorithm to select individuals for a range of collection sizes, 6 to 16 in the table, and I just show Rhodamnia as an example. And if we look closely at the genetic relationships in this tree for n equals 6 and 10, So 6 is in the blue circles, 10 is in the green triangles. So we can see that the selection is scattered relatively evenly across that phylogenetic tree, which reflects the fact that genetic variability is distributed along a latitudinal gradient. So this selection approach can be tailored to the goals and design of specific conservation actions. So currently we have the myrtle rust assays going on for rhodamnia at the Plant Breeding Institute at the University of Sydney. And also we have whole genome sequencing, so 40 samples from each of the species going on, and the analysis is underway. And we also have some other projects with the other Myrtaceae species with variable resistance. So hopefully they will be able to understand the genetic basis of resistance and then help optimize conservation populations for genetic diversity as well as rust resistance. And building a framework for a coordinated response which encompasses conservation genomics is crucial for the management of myrtle rust impacted species. And I'd like to thank everyone that has been involved in this myrtle rust research, as well as the funding bodies that have made this work possible. Thank you for listening.
Thanks to Stephanie for a really great example of how we can take genetic information and utilise that in our ex situ collections in gardens. Our next speaker for today is Dr. Gemma Hoyle. Gemma is a seed scientist responsible for designing, carrying out and reporting on research projects at the National Seed Bank. Her work focuses on seed germination, dormancy and longevity to improve the conservation and management of Australia's native flora.
Morning, everyone. I acknowledge the traditional owners of this land and the Ngunnawal people where I work and pay my respects to their elders, past, present and emerging. So my talk today is about the management of conservation seed bank collections. Essentially what I'm going to talk about is next steps in conservation management when seeds don't germinate. Luckily for us, it would seem that seeds of most Australian plant species have the potential to remain viable in conservation seed banks for hundreds of years, including safely stored with us at the National Seed Bank in Canberra. But there's very little point, as we've heard a lot this week, in collecting, processing, drying and storing seeds if we're not able to germinate them when it comes time to pull them out of the seed bank. Seed banking is not just about putting seeds in a freezer and closing the door. We need to be able to use collections to propagate plants in the future and to carry out seed-based research. And not just that, but we need to be able to monitor viability of collections over time. And since germination is the best indication that seeds are alive, we do that with initial and ongoing germination testing. When we pull seeds out of the bank, obviously we aim for 100% germination. But experience has taught us that the quality and the dormancy status of wild seed can vary significantly. Not to mention that there's a huge volume of collections to manage. The National Seed Bank adds around 300 new collections a year. Therefore, we've come to recognise anything above 75% germination as a success and sufficient enough to declare that no further investigation is warranted. And we call this a germination pass. But often, as you would know, achieving 75% germination is easier said than done. For example, this figure shows 37 previously untested accessions along the bottom and maximum germination in controlled conditions on the y-axis. And the dotted line is that 75% threshold. And as you can see, there's everything from 100% germination down to 0. And only 8 of these collections achieved a germination pass. These collections were of threatened grassy ecosystem species collected in Southeast Australia. This map shows the distribution of major grassy vegetation subgroups in Southeast Australia prior to colonisation. And this is what the map looks like today. Grassy ecosystems are among the most degraded and altered vegetation communities here in Australia. The natural temperate grassland communities pictured here were listed as critically endangered under the EPPC Act in 2016, and then in 2020 they were upgraded in the ACT to critically endangered as well. The remnants that we have left are home to more than 300 different species, and the National Seed Bank holds collections of at least 55% of these. So given their conservation status, the collections highlighted here certainly do warrant further investigation. When seeds don't germinate enough to achieve a pass, we ask ourselves, is it because they're not viable? We can investigate viability using the tetrazolium chloride test, or the TZ test, and I'll talk more about that in a minute. Is it because they're dormant? This is when seeds have evolved to postpone germination, which is very beneficial for seedlings in the field, but it's a hindrance to our work in seed banks. If we suspect dormancy, we can investigate by applying various seed pre-treatments. Or is it because seeds are empty? We can investigate seed fill using X-ray analysis. And collectively, when we assess results across all these investigations, we can make informed collection management decisions. And that's what we aimed to do, assess the viability and germinability of these grassy ecosystem seed bank accessions, and assign each one with a next step towards achieving a germination pass or understanding why we can't. Today, I'm going to focus on the seed bank collections, but they did include 29 different species, and I'm happy to discuss species with anyone interested afterwards. All the collections were sown in control conditions that were designed to mimic those that seeds would experience in situ post-dispersal. And here's a reminder of what those results looked like. So next steps for those that achieved a germination pass include rebanking that collection and confidently utilising that seed with their known germination requirements. But what about the other collections? There's growing evidence that many grassy ecosystem species respond to fire-related germination cues, as we've heard this week. Where we had enough seed, some of the collections were given a pre-treatment designed to mimic changes that may occur in the field during or after a fire. The TZ test was used to estimate the viability of the collections that didn't achieve a pass. We used fresh samples of seeds, not those remaining at the end of a germination test. Seeds were chipped, soaked in the TZ solution for 24 hours, and then dissected and examined. Living embryos usually stain red, while non-viable tissue doesn't stain. I know the TZ test is considered subjective, time-consuming and sometimes misleading, but there are guidelines available for a lot of Australian plant families. And in the absence of germination and the context of managing conservation collections and diagnosing dormancy, I do think it can be a useful tool. Also, most people that use it, well, the more people that use it and validate it or adapt the methods for different species and report that, then the more confident we'll all get using it. And then we also X-rayed collections that achieved less than 20% germination, and we calculated percentage seed fill for these collections. All the stats were performed in R, so we compared the TZ test results with maximum germination achieved with or without a pre-treatment. And we used the 95% confidence intervals to explain this difference and enable us to interpret the nuances between collections rather than a simple, yes, they're different, or no, they're not different. And we also assessed the overall effect of each fire-related pre-treatment on final germination relative to control. This table describes the 6 categories into which accessions were placed. Don't worry about reading it all. Just notice that we've got next steps numbered 1 to 6, which tests were done and their results, how we interpreted these results following analysis, and the actions that we would recommend. In particular, steps 2 to 6 are all the scenarios in which seeds achieved less than 75% germination. So let's look at the scenarios one at a time. Next step number 2. If you look at these examples of confidence intervals, they all contain 0, and the lower limit is above 20%. We interpreted this as there being no significant difference between the germination and the estimated viability. So collections had achieved a pass relative to their estimated viability, and we call that a viability-adjusted pass. And we can recommend rebanking these collections and using them with these known germination requirements. Next step, has it jumped? No. Next step number 3. The confidence intervals here again contain 0, but the lower limit is below -20%. So we interpret this as there being almost no significant difference between the two tests. And here we would recommend prioritizing these collections with repeated germination and TZ testing, using an increased number of seeds per replicate. This would enable more robust analysis of the results and likely give us a clearer next step one way or another. Next step number 4. This was where the confidence intervals lay entirely below 0. We interpret this as viability being significantly higher than the germination achieved. So these results suggest that there's a lot of germination potential in these collections. 11 collections fell into this category, and these really interest me because it's worth investing time and resources into investigating germination strategies and possible dormancy mechanisms here. And we would do this by investigating germination response to things like dry after ripening, stratification, gibberellic acid, scarification, etc. It likes to jump, but sometimes it goes back. Can you see next step number 5 or 6? 5. Next step number 5, we're nearly there. This was where both the germination and the TZ tests yielded less than 25%. So we deemed analysis wasn't worthwhile here, and we'd recommend validating both these test results by investigating alternative TZ methods and different germination test conditions. And lastly, next step number 6. This was where, unfortunately, there wasn't enough seed for a TZ test after we'd done the germ testing. So analysis wasn't performed because conservation collections are just sometimes not that big. And here we would consider recollecting this species, particularly if we don't have other collections of this species in the seed bank. 4 collections would benefit from further processing to reduce the empty fraction and increase seed fill to at least 85%. Empty seeds are not necessarily a huge issue for us since they are removed from the equation when we calculate percentage germination. But empty seeds do waste curation resources. They attract mold in germination tests, and they just have us thinking that a collection is bigger than it actually is. Remember those fire-related treatments? This figure shows germination in control conditions in the red bars, and then germination following various seed pre-treatments in the blue, pink, green, and purple. And basically a bunch of collections achieved a pass, not in control conditions, but following one of these pre-treatments. Obviously, the number of tests that are possible rests on the size of the collection, as well as whether or not we have multiple collections of that species and what options we have for recollecting or replenishing a collection. Following on from this work, we've been able to use some of the larger collections that responded to fire cues in investigations of different types of smoke water. And we've also been able to use collections that achieved a pass in comparative longevity studies that rely on high germination. So in summary, being able to germinate seeds of each accession is critical to effective seed banking for many reasons. When germination isn't sufficient, we need to make sure that we're allocating time and resources to investigating germination of viable and potentially dormant seed rather than dead or empty seed. The analysis and interpretation described here seems to suit our needs. Other seed handlers could be more or less generous with their boundaries around that difference between estimated viability and germination. And I'd be interested to hear how other people estimate seed viability and how they differentiate between dead and dormant seed when germination is low. We're grateful to the Mundango Charitable Trust for funding this work and the NSB staff and volunteers for helping with the testing. Thanks to the team at the NSB for just being awesome to work with. And thanks to the ANPC for this excellent conference and for having me talk today. Thanks.
Thanks, Gemma, for taking us through some collection management examples, and I think there'll be good discussion in the Q&A session. Our next speaker is Kathryn Scobie, and Kathryn is a nursery horticulturalist at the Australian National Botanic Gardens in Canberra. She manages the permanent potted nursery collection, which consists of hundreds of vulnerable and threatened Australian plant species from a wide range of genera. Thanks, Kathryn.
Morning. Today I'd like to focus on sharing with you how we support conservation work at the nursery at the Australian National Botanic Gardens. So at the ANBG we hold 24% of the plant taxa listed under the EBPC Act. That's within our nursery, within our glasshouses and within the living collections in ground. And then the National Seed Bank have additional collections on top of that 24%. So our collection represents species from all over Australia, as well as some of our island territories like Christmas Island. So you can imagine within the Canberra climate that that's a challenge at times. Our collection is also a scientific collection, which means that for most of the plants within our collection, they're accessioned. They have a unique identifier that tells us information about that plant, which at best gives us the details of when that plant was collected, who did that work, the propagation history, what its history has been like in the nursery, and links to the herbarium specimens. We've had a strong increasing focus on plant conservation at the ANBG in recent years. And one key example of that is that the nursery sits within a dedicated new conservation team with our own conservation manager. So we're moving in an exciting direction. Our nursery facilities are pretty great, I can't lie. We've got 5 tropical glasshouses, 2 large polytunnels. One of those polytunnels is dedicated to the permanent potted collection that I manage, and the other one is our primary plant production area. We've got 7 small poly prop houses and also purpose-built potting shed. We've got a shade house and hardening off areas as well. So lots of space to do this good work, although sometimes it feels like never enough space. Just want to take you through this video which shows great linkages across the gardens in conservation context. Don't know if we've got some audio.
Australian plants. It's also an internationally renowned scientific institution. And behind the scenes is a vast array of research, discovery, and conservation work that plays a vital role in conserving our native flora. Here at Black Mountain, the National Seed Bank, the Gardens Nursery team, the Living Collections team, and the Australian National Herbarium work together to research and document Australia's biodiversity, to study and maintain plant populations, conserve rare and threatened species, and provide specimens and information for scientists across Australia and around the world. Their close links and specialist skills and expertise put the Australian National Botanic Gardens in a unique position to help study, conserve and secure Australia's threatened species, now and into the future. You can find out more at anbg.gov.au.
So I'd like to take you through just a couple of case studies of current projects that we've got going on in the nursery at the moment. There's a lot of exciting work. The first one I'd like to start with is a Survive and Thrive partnership project that's been developed to protect bushfire-affected species within our subalpine areas, so Namadgi National Park in the ACT, Kosciuszko in the New South Wales, and Alpine National Park in Victoria. At the nursery, we're targeting 5 key species with 7 secondary species. So we're looking at Amelia capitata, Viola imbricata, Leptospermum namadgiense, Alaria rhizomatica, and Leonema lamprophyllum subspecies obovatum, which you can see pictured there on the slide. So this project is combining expertise in seed biology, conservation and horticultural research. And it's a really unusual one for us in the nursery because our goal isn't actually producing plants. What we're doing is producing the research on how to successfully propagate these plants for future rewilding projects and seed production work, and to share this research with other people. So this is an amazing project for us to work on because that type of outcome is usually a byproduct of the work that we do every day producing plants, but the information tends to get lost in our collective memories or deeply hidden within our propagation databases. Another reason this project is particularly special to us is because we were able to inform the project design from the beginning of this project, which is a powerful position to be in. It's also multi-year, it's also fully funded, so gold star from us on this project. Swansona recta is another species that we've been doing a lot of work with over the years. So this little pea is endangered in listings in New South Wales and Commonwealth. We're growing hundreds of plants for translocation at a number of different sites and working with Central West LLS, Central Tablelands LLS, and also the ACT government on this one. Depending on the day in the nursery, Swansona recta can be a dirty word because while this plant is definitely a stunner, it's also quite the diva. It can be difficult to germinate. It only particularly likes germinating with friends, not in individual forestry tubes. So that's a challenge for us in pricking out many hundreds of seeds from germination trays. It likes to have a special mix. It requires a longer growing period before it successfully transplants out into translocation sites. It's fussy about the water that it needs, and also because we are translocating this plant to a number of different regions and we're working with different lineages, anytime we get flowers in the nursery, we need to snip those off to make sure that we get no crossing of lineages between the plants that we have. So tricky, but also beautiful, so we forgive it. Eucalyptus recurva is another species we've been doing quite a lot of work on over the years at the AMBG nursery. This is a critically endangered species in New South Wales and Commonwealth listings. There are only 6 plants of this species surviving in the wild to our knowledge, and a number of those are in pretty poor habitats. There's not great seed set in the wild. We've had low propagation success with this one, so we're really focusing our efforts on grafting. If you look closely at that image there, you can see a little graft union in the middle there, so that's fairly delicate and specialised work. And our end goals are growing plants in our seed production area, but I think we're still quite some way off achieving that. We partner with the New South Wales DPE and also the Office of Threatened Species Commissioner on this one, and have recently been able to collect some more materials, so we've got some grafting happening right at the moment. It's also been instrumental in driving some upgrades to our facilities to try to get a really nice high humidity enclosure to help us support those plants in that really vulnerable stage when they're grafting at such a small size. Okay, we got there. Muehlenbeckia tuggenong, this is probably the closest species physically to where we work on. It's endangered in the ACT and Commonwealth listings as well. We've held this in the nursery for quite a long time, and it's one of those plants that you can't seem to kill. Which is fantastic for us. It's very low care in the nursery, but unfortunately it doesn't seem to have the same longevity out in the wild. And as such, we now have plants within our collection that we believe to be now extinct in the wild. So we are focusing on building up lineages and propagating fresh material and partnering with the ACT government on looking at ways that we can restore the wild population of this plant. Just a few other projects that I'd like to touch on that we're towards the end of. We have been working on an Epacris microphylla translocation. We have also been doing quite a bit of work on bushfire-affected species from Mount Imlay down in the far south coast, working together with our friends at Buddery and also New South Wales DPE. Our Trumps project, you may have seen a poster on some Trumps work that's been going on in the seed bank, and certainly we've heard some other talks about study into Trumps seed. So in the nursery we've been growing plants for these, some of the species in these highly restricted environments on tropical mountaintops that are facing potential catastrophe as climate change progresses. So we're trying to find out as much as we can about these plants and how they like to grow while we still can. The Scarlet Robin SOS project is pretty much wound up. It was a nice little one in that we were able to combine, kill two birds with one stone. It's probably not an accurate thing for our Scarlet Robin project to say, but we were able to select species for habitat corridors for this project, and as part of that we were able to put a couple of conservation species into that mix. So that was something nice that we could do there. We also produce plants in the nursery for scientific research, so we work quite closely with various scientists at the CSIRO. So we produce plants to study their sensitivity to phosphite as a phytophthora treatment for various genomic sequencing and also for taxonomic research. Okay. Conservation in the nursery can be just simplistically represented here by material coming into the collection, us propagating it growing on, and then trying to secure that conservation collection. And it's all surrounded by collection management so that we are keeping accurate records and doing the right thing in securing these important collections. And each stage of this process can look really different depending on the context. So you can see a number of different representations of material collection here. So it can be material coming in from external field trips, it can be us wandering out into the gardens and collecting material, or heading up into one of the glass houses to collect something growing in the nursery or from our permanent pot collection, or having seeds come up from the National Seed Bank and working on those. So here we've got shots of looking at Phibalium squamulosum subspecies Ozothamnoides in the field after bushfire. Not enough material there to actually collect, but at least we know where it is, we know it's coming back, and that's there for potential future collections. We've got bagging of seed of our favourite Swansona erecta for future seed collection from within the gardens. And then Pete there collecting some cuttings of Eucalyptus recurva. And when we get into the propagation phase, that also looks very different depending on the context and the species that we're working with. And there are so many variables that can affect the success that we experience. from the media that we use, the watering regime, the environmental conditions in the propagation houses, what hormones we choose, pests, the season, what condition the material is in when it comes to us, and on and on. So we have a great database that we maintain of our past propagation attempts and success rates. So if it's a species that we have experience with, we're in a good position to make incremental improvements to our propagation success each time. If it's something that we haven't worked with before, then it's open slather to make some informed decisions about our initial approaches, and then we continue that experiment moving forward. So, 1 minute. Okay, Pete's grafting up in the top left-hand corner there. We've got trays of Swainson erector seed all coming up as babies. On the top right, Leptospermum nemagiense cuttings and some germinants that have come up to us from the seed bank, so a range of different propagation there. The growing on phase also looks really different. We've got a Pacris microphylla ready for translocation in forestry tubes up the top left, some Hakea pulvinifera, one of my water dragon friends in our permanent pot collection on the top right there. We have some Nematolepis rididophila fruit that was the outcome of a pollination exercise that we did, and just Beck there doing some routine potting on. So lots of work goes into the growing on phase. And securing conservation collections looks vastly different and should be multifaceted as well, with ex situ and in situ where possible. So we have our seed production areas. We have Lamessia tasmanica sitting in a shade house within a shade house, trying to give that optimum conditions. We also share plants with other botanic gardens, so that's some Tromps plants going off to Cranbourne in the bottom left-hand corner, and then plants in our conservation garden. Lots of risks and challenges at each stage, and I guess the key takeaway here is working together with project managers to understand what those risks and challenges are, and to try to work together to mitigate them as best as we can. And these risks are real. These photographs are taken within 12 months on site at AMBG. Weird things happen in the nursery every day, from wind damage, our annual armillaria burn, and just rat damage on a wire that controlled environmental conditions in one of our glass houses. So all sorts of things go on. Last slide. What I would just say is, I know I'm banging a well-beaten drum from this conference, but I'd just like to reiterate some key thoughts on ideal conservation projects. We know that more funding equals more people and more capacity to maintain collections. Having realistic requirements just means that we're all going to be able to achieve the same goal, and more project time particularly is a really big one, helps us achieve great outcomes. Botanic garden staff, nursery staff really want to do this work with you all, and we look forward to loads more of it in the future. Thank you.
Thanks, Kathryn, for a behind-the-scenes look at what happens in a nursery. Our final speaker for the session is Dr. Peter Cuneo. Peter's the manager of the Seed Bank and Restoration Research at the Australian Plant Bank, where he leads the statewide Threatened Species Seed Program. Peter's main research interest is in restoration ecology, including threatened plant translocations, management of invasive species, and restoration techniques for grassy woodland ecosystems. Thank you, Peter.
Thanks very much. Firstly, I'd like to acknowledge the traditional owners and pay my respects to the elders, past, present and emerging. The projects I'll be talking about today actually occurred in Darug country in Western Sydney. So today, I just want to go through a number of translocation projects we've been working on at the Australian Plant Bank over the past 5 years, and just really reflect on our experiences and the successes and some of the failures as well, and just to see how we've gone with these projects, particularly in the hard Western Sydney environment. Well, there's probably easier areas if you want to make biodiversity gains in Western Sydney, but having said that, 13% of the vegetation still remains, and we have 22 threatened species and 12 threatened ecological communities. So it's amazing what still exists in Western Sydney, and these small pockets of threatened species can still exist. Of course, Western Sydney has really borne the brunt of urban expansion over many decades, and we've got significant urban development footprint, and of course we have major infrastructure projects now. For Plant Bank, we're located right in the thick of it, in the middle of the Cumberland Plain. In fact, we have a lovely Cumberland Plain woodland remnant right outside Plant Bank. So it's given us quite a bit of experience in working with Western Sydney species, and particularly their ecology. We tend to get the phone calls from a whole range of people. Sometimes they go along the lines of, hey, we've got this endangered plant and we're about to start construction in 4 weeks. What can you do for us? And so, yeah, they're quite difficult, and they're the sort of phone calls that I guess Greening Australia and some of the other partners tend not to want to touch. So we're talking, petunias, like Nathan was talking about today, terrestrial orchids, all those sorts of difficult things. We're always keen to take them on. So the two projects that I want to talk about today are Parmelia spicata, the spiked rice flower, which is it's part of the Western Sydney International Airport project, and the other one is Herbertia puberula subspecies glabrescens, the Bankstown Herbertia, which is part of the Saving Our Species program. So the Parmelia is a little low-growing understory plant endemic in Cumberland Plain, and the Herbertia is again a prostrate plant, only occurs at one location, at Bankstown Airport. So I'll start with the Parmelia. This is the sort of projects that we're getting in Western Sydney. The airport, it's a massive project. I don't know if anyone's driven on the Northern Road, just the scale of it is extraordinary. $5.3 billion project, opening in 2026, and they're moving 20 million cubic metres of earth, so the entire landscape is being transformed, the entire hillsides are disappearing. Whenever we drive out there, we get totally disorientated. But yeah, it's a massive project. One problem with the Western Sydney Airport project is that there was a major population of Parmelia spicata, 3,000 plants found right where the runway was going to be constructed. So we got engaged through GHD, some consultants to the federal government, as part of a biodiversity offsets program in 2017. So we started off with some seed collections, always a good place to start when you know a population is going to disappear, and started a propagation program at the Australian Botanic Garden, just trying to understand the propagation, some of the ecology of Parmelia, and several other species as well that were impacted in the airport footprint. Maurizio and his team did a conservation genetic study, which was right across the Cumberland Plain region. So it gave us a bit of some really good information of the populations that were there and the genetic structure. And of course, this all led to a translocation plan that we wrote with GHD. But any of these big projects, really complicated project structure. So you've got federal government, you've got numerous sub-consultants. So yeah, it was quite complex, just all the administration, that's for sure. So how did we go with the Parmelia? Well, they were hard won, but we got over 3,000 seeds. So it took us multiple seasons to get those collections. You can see the mesh bagging there. But it did take quite a number of seasons, and we kept being impacted by drought. In fact, the plants you can hardly see above ground level for many years. So we did put everything together. We've got the genetic study, we've got a propagation plan and study, and the translocation plan. So we've got those three key elements. As part of the project, we also developed an ex situ potted collection of targeted genotypes. So this was, of course, informed by the genetic study. So we now have a very good resource for future translocations. And with that potted collection, we were able to really target the genotypes and minimise the number of plants we've got in the nursery, yet maximising the genetic diversity held. Well, I have to say, our beautiful translocation plan has not been yet followed in detail, but I guess as we move to the next step, we're hoping that that will be implemented. There is an environmental conservation zone as part of the airport, and plants will go into there as part of the management of that area. Because the plants were going to be lost, some plants were actually salvaged. This is where different contractors came in. We lost a bit of control on the project there, I'd have to admit, when the bulk earthwork started. But they did actually dig some up and translocate into the conservation zone with apparently 90% success, which is quite good. One of the fun things about working with plants like this, you can see that photo there of the lignituber. When plants are going to be destroyed, it gives you a bit of opportunity for some experimentation, and you can actually dig one up, and you can see that amazing lignituber there, which is very typical of a lot of the Cumberland Plain species, and probably makes it an ideal candidate for actual salvage and translocation. And in fact, Greening Australia actually dug some up, and they've got them in their seed production area in Western Sydney as well. So not all was lost. So what are the key learnings we take away from this? We've had plants produced, but we didn't have an initial conservation goal clearly defined. But now we do, as part of the translocation plan. You've got to be prepared for the long haul with these projects. We've been working on this since 2017, and you have to be very adaptive. As I said, you allow multiple seasons for the seed collecting, very important. And of course, with these big projects, the admin complexity and the site access is really complicated sometimes with WHS, and you've got big machinery moving around. Potted collections, it was interesting hearing about some of the potted collections at ANBG. They tend to sort of almost like keep growing overnight, I think, expanding. You've got to really watch those because they can get very large and take a lot of time and effort to manage, and so that's got to be built into the contract, the long-term management of those conservation resources. How did we go with the Pimelia? I think we're probably maybe not gold standard, but maybe silver, but we'll get there. Bankstown Herbertia, this is the next one. This is a really interesting one, critically endangered, small population persisting in a highly urbanised environment. So it's actually growing right next to, in a paddock, right next to Bankstown Airport. And it's just been managed by high slashing for many years. But it persists there quite happily. Endemic to the Sydney region, 70 plants there. So this was identified early as part of the Saving Our Species program as a plant that could be lost, and we had to establish some new populations. So being one of the first SOS projects, we set out to do a gold standard translocation plan. We had good lead time, we had good funding, so we set about doing that. Again, part of that design was a genetic study to design the new populations, and of course to maximise the genetic diversity and minimise the kinship, because you haven't got a lot of diversity in a population of 70 odd plants there. You can see there the photo, Jason Bragg from Maurizio's team just We're just trying to sample and look at clonality as well, which was also a big issue. Of course, when it's growing in a paddock next to the airport, you say, okay, well, what was the original vegetation type? Where's the translocation site going to be? What is it going to look like? So there's a lot of soil testing and looking at, I guess, forensic work, as it were, just trying to understand the original vegetation type. And so we decided it was sort of going to be Cooks River clay plain sort of scrub, probably around the Georges River. So multiple planting sites located, and then we set about propagating 28 genotypes at the Australian Botanic Garden. So how did we go? Well, we had we did achieve gold standard in one of the spots at the Voyager Point site, 94% success rate. And the plant started to flower and set seed. Everything was going perfectly until the Black Summer fires, with the help of an arsonist out from Liverpool. Our entire main population was destroyed in January 2020. So that was a huge setback for the project. The other sites, we had some other sites, fortunately, where we achieved 64% and 56%. So I guess that's the emphasis, having multiple sites. We haven't seen any seedling regeneration at the Voyager Point site. I suspect there hasn't been enough accumulation in the soil seed bank. So we're back to square one on that one. One of the sites, we've actually used some seed-grown plants as well, and they're doing quite very well. Some of the interesting observations as part of this project, and they're all different. Some of the genotypes were difficult to propagate. So the nursery, we had to keep going back to individuals in the field. They are very hard to strike from cuttings. So obviously that's good. You're picking up genetic diversity, but very recalcitrant, I guess, for propagation. But interestingly, once we did actually propagate these, they established quite strongly in the field. So again, pretty confident we're capturing some good genetic diversity for this species. The seed-grown plants, as similar to what Nathan's experienced, the seed-grown plants were more robust and established very well. And that's an interesting observation. It produces a dilemma, really, because the seed-grown plants, of course, the genetics are less known. Multiple sites to spread the risk. And interestingly, the plants don't like competition or shading. We assume they would have been part of an original forest structure originally, but they tend not to like the shading or competition. The public-private partnerships with contractors and other individuals, councils, has been really important for this project. And of course, it's important not to give up easy, even when your main population has been burnt after 3 years of planning. Conclusions. Well, genetics is really a valuable tool in designing these populations, really important, and right through to the actual, even to the potted collections. It's allowed us to get them down to quite a manageable size, which is really important, and so we can have a really good cutting grown genotype collection for future translocations. But always in these projects, never underestimate the importance of horticultural expertise. It's always really key. You can have all the genetic information and everything in the planting, but the skill in propagation, the skill in the planting and the maintenance is really critical. And here you can see Mark Weiler, our head propagator, actually on site. He's grown the plants from cuttings and he's actually there on the planting day as well, just helping out and advising. Really important. Is there a role for seed-grown plants? Well, I'd like to think so, but obviously a lot of the translocation plans prefer genetically selected cutting-grown plants. So I guess that'll be an interesting debate, but certainly the persistence of seed-grown plants looks significant. As we all know, multiple genotype plantings are great. but they're record keeping intensive. So when you're growing 28 in the nursery, it's a big project. Potted collections are resource intensive, absolutely. So how do we go all up? Well, I think, we're probably, with this one, we're probably at a silver standard, but we've got more propagations coming through, and we're still planting this autumn. But I guess the real take-home thing from a lot of these Saving Our Species projects that we've worked on is we now have a very good integrated model across RBG. So we've got the genetics, we've got the seed collections, we've got the seed germination capability, and we've got the nursery production capability, and as well as plant disease diagnostics as well. So it's helped us to sort of bring together, I guess, under the one roof, a really nice model for translocations into the future. I'd just like to thank my colleagues and project team, RBG, and at DP, Ahmed Sharif in particular, as the project leader. Thank you very much.
Thanks for that, Peter. Some examples of quite challenging genera to work with, so lovely to see such good success and the way the program's developed. So we're now going to go to a Q&A session. So I'm sure you've all been thinking of lots of questions while the speakers have talked today, and open to the floor for anyone who has a question, but I'll also check with Amelia for online. Shall we start with online while the room are thinking?
Sure, yes. So Nathan, I have a question for you from Steve online. He's asking if you have any ideas about the seed dispersal mechanisms of Petunia.
Yes, we do. That's a great question. Thanks. So, due to the heavy-weighted, fleshy drupe dispersal unit of Petunias, they're predominantly dispersed via gravity, and certainly when we do see recruitment in the wild, we see the majority of that directly sort of around the adult plant. That's not to say there isn't any long-distance dispersal. The fruit are consumed by birds in particular, other small mammals. They do provide a valuable resource in an otherwise sometimes highly depleted resource environment. So we do see those long-distance dispersal events. They're just not as common.
Thanks. Martin's got a question.
Yes. One question, two parts, to no one in particular. In the Healthy Seeds program, one of the things that came up time and time again with collectors in the field was very few collectors and or merchants ever do seed testing or asked for seed testing. And they often don't. I only know of one that seems to have good capacity to do that. And the issue has been raised a number of times by these people of in the opportunity for seed exchange when they do seed collections of particularly rare things that they're doing, exchanging and getting seed testing with various botanic gardens, etc. That opportunity, but also using the network that's out there for assistance with things that they're having trouble with germinating, and also the opportunity for these people that are out in the field all the time and often know where things are, how we can capture that capacity and communication with the people through the research organisation. So sort of multi-parts there, and yeah. Is it realistic even? That's the other thing.
Maybe we can, don't know, the panel don't, oh, Peter, were you about to answer, were you? Go ahead.
Thanks for that multi-barreled question. I guess in germination testing, yes, agree, it's a really big issue for the native seed industry. I guess one way we might be able to approach that is if we can have develop the more quick and dirty style germination basic tests may help, because I think we all understand that they're not going to have laboratory facilities like Plant Bank or ANBG. Maybe there's a halfway house that we could, us as scientists might want to think about how we can get just some basic germination data. I agree with that. Just trying to get the second part of your question, sharing information. Well, speaking on behalf of the RBG, I guess we've probably got a long way to go in terms of having our germination data out there. I mean, there are other good sources. Kew, a lot of the Australian seed germination data, is held through Kew. So that's a really good source for basic germination information, particularly on species that might be difficult to germinate. In terms of sharing information, again, a lot of our records are on the Australian Seed Online and ALA, if we're talking just distributional data. Certainly hard to set up a mechanism there. For us, we often use regional consultants when we're working in areas we're not totally familiar with, and quite often that will lead to private landholders as well, like as our local connection. So I guess it's really a bit of a two-way street there between government organisations like ourselves and the industry. But I guess the flip side of that is there'd be a lot of collectors out there would have their nice little patches that they could go and collect every year and they wouldn't want anyone else to know about.
The other thing is that they're not talking to you large collections. They're sort of sub-samples of things that they're getting, and they see here's an opportunity.
Sounds like there's some great opportunities for building connections and working through ways to build that. And perhaps we can keep that discussion going over morning tea. I just want to make sure we get any further online questions or others in the room. Anything online, Amelia?
Yes, we do have one question, and it does go into the detail of seed testing. So Gemma, it was a question for you from Meg Hurst. She's interested in how you're incorporating tetrazolium testing into your germination protocols. And I'll just add a comment of my own. There's a fair bit of information about both basic seed testing and quite complex germination protocols between the Flora Bank and the germplasm guidelines, including that tetrazolium testing.
Yeah, thank you, Meg. I guess I just want to make the point that, yeah, it can be a time-consuming test to do, which is why we don't routinely do it on every collection that comes in. It is quicker and easier to do a germination test routinely with everything that comes through, but when you're not getting that germination, when you've given the basic germination testing your best shot and you're not getting that germination, then it's those collections that we would try to target for a TZ test, which is a 3-day procedure, but you can get quicker and more efficient at it, and there's lots of guidelines out there now. I know that it began with just crop seed guidelines, but that's extended now to a lot of Australian families. So you can run a bunch of TZ tests at the same time within a week. And it really just opens up, I think it's a good tool. And opens up possibilities then for whether or not you're going to invest your time and energy on looking at more complex germination requirements or trying to alleviate dormancy or not. Because often there are non-viable seed in our collections, no matter how good we are at collecting seed, and that's just the case with wild seed often.
Thanks. I think we've got a question here at the front of the room.
Thanks for those great talks. Collectively, you all hold a lot of knowledge about propagation, and I'm just wondering where you share that knowledge, or is there a database that we can access to learn more about propagation?
Sorry, I'll just jump in very quickly. I know I'm not formally on the panel. In the new edition of the germplasm guidelines, which are a free download and also available on the registration desk, we've added a whole new chapter in this edition about the role of the nursery, propagation protocols, a whole double-page spread about cuttings and other types of propagation. So there is some information there, including down to species level for some, and I'll also let Kathryn answer.
So we have a database that's actually available online that I can share the address for, and it's not the most user-friendly interface, unfortunately. So you really kind of need to know what you're looking for, but that's what we use, and that's what our database talks to. So, but there's a wealth of knowledge in there, and I think it would be a great project to see someone put that together to share that more broadly.
I think we had the question down the front might have come up first.
Yeah, question to Gemma. I was just wondering about the stratification involving different day lengths, different amounts of daylight as opposed to night time at different times of year, and also different temperature regimes. You know, we have simulating winter conditions and summer conditions. How do you actually account for that stuff where that's important? Is it that important?
Do you mean like germination incubator temperature regimes?
Yeah, so you know, having conditions which simulate winter conditions as opposed to summer conditions, so day length differences and temperature differences. How do you actually take that into account?
Well, we start by looking at when in the year the seed is dispersed naturally, and so what that viable seed is going to experience in situ straight after it's dispersed, and we would start there with trying to mimic those temperatures and those light conditions in the incubator. But some seeds have evolved to postpone germination until after the winter, or after a rain event, or after it's been eaten by a cassowary or whatever. So when seeds don't germinate in those initial conditions that we've given, where we've tried to mimic what they're dispersed into, we then start to look at, okay, well, what do they experience next, and we might tailor our treatments to a winter chill in the dark under the snow for 8 weeks or something, and then put them in back into the same germination conditions that we've already tried, and often that will do it because they want to postpone germination to give the seedling the best chance in the field. So does that answer your question?
So I can see that there are so many questions in the room, but I've done the worst job of chairing by being too enthralled with the discussion, and we're very over time, and we'll not get morning tea if we don't wrap up now. So I invite everyone who's still got a question, please find one of the speakers and ask it over morning tea. And one last thing is just to thank all of our speakers. So thank you everyone for a great session.